Okay, we'll move on to Chapter 6 now, where we'll talk a little bit about microbial growth and some of the factors that are involved in microbial growth. Microbial growth basically is the increase in the number of cells, but not necessarily the cell size. Microbial growth basically refers to the number of cells, not to changes in size. Colonies are basically the accumulation of cells large enough to be visible without a microscope. Now, this will be important to you, again, if you're going into any type of clinical setting where colony counts are important in being able to identify microorganisms that may be growing within a particular environment. Some physical requirements that are a part of growth. Temperature. We talked about temperature as it related to enzymes. Remember, enzymes function optimally at certain temperatures. Temperature, again, the minimum growth temperature is the lowest temperature at which species will grow, and the maximum is the highest temperature these organisms will grow. The optimum growth temperature is that which the organism will grow the best. I think those terms are pretty self-explanatory. Again, when we talk about temperature, there's minimal growth temperature, optimum growth temperature, and maximum growth temperature. You can see the categories of microorganisms and where they may grow at best based upon temperature here reported in Celsius. Psychrophiles obviously growing on the lower end. When I say obviously, it's just because the illustration is showing them on the end here with extremely low temperatures. Basically going to the extreme, we can see microorganisms that grow at extreme temperature ranges, the hypothermophiles. And then all the organisms in between. Psychotropes basically grow best between 0 degrees Celsius and 20 to 30 degrees. We see these organisms are the ones responsible for food spoilage. So even in a refrigerated setting, these organisms can grow. We can also see these organisms are capable of growing in extreme areas in the environment. Microorganisms have been identified in the polar caps, these regions that fall in these categories. Many microbiologists will define psychrophiles as having optimal temperature growth of about 15 degrees, usually found in the oceans and, as I alluded to, the Arctic environment as well. The term basically refers to as psychrophiles, basically moderate, sometimes or facultative psychrophiles. These have been proposed as organisms that grow well in refrigerated temperatures, but they have an optimal temperature growth as well, as most of these organisms do. In these terms, we see these organisms again causing food spoilage as well. And the illustration pointed out in your book just demonstrates the rapid growth of bacteria, and some of the organisms also will produce a toxin that's associated with them that again can survive at some extreme temperature ranges. Mesophiles basically are the most common of the microbes. They have an optimum temperature, 25 degrees to 40 degrees, and obviously that's very consistent with our immediate environment. So again, these are the most common microbes that you're going to see in the environment. Thermophiles basically are capable of growth at high temperatures. Many of them have optimum temperatures of 50 degrees to 60 degrees Celsius, and many of them are capable of growth below 45 degrees. Now, hyperthermophiles, basically extreme thermophiles, found among the archaea organisms, and they have an optimal growth range of about 80 degrees or even higher. There are also organisms that live in specifically extreme and or moderate pH balances as well. Again, pH referring to acid-base component of an environment or a solution. Bacteria can grow between a pH of 6.5 to 7.5. Molds and yeast basically grow through a pH of about 5 to 6. And acidophiles grow in an acidic environment. So again, pH extremes can also be a factor in either promoting and or inhibiting bacterial growth depending on the type of organism, well I said bacterial, microorganism growth, depending on the organism and depending again on the environment. Most bacteria actually grows best at a narrow pH range, just as humans do. Our normal pH is 7.35 to 7.45. We see bacteria can exist in a range of about 6.5 to 7.5. And what that tells you is, look at that wide range of variances that bacteria can exist in versus the human biological system, which really only can exist within a very narrow range. Very few growth is between a pH of 4. However, many bacteria such as acidophiles responsible for acid fermentation are markedly tolerable to acidic environment or acidity. The buffer is sometimes added to a medium to neutralize acids. Examples of buffers are phosphate, salt, peptones, and amino acids that can be used in solutions to again neutralize the acidity of a particular substance. This is important again when you're trying to inhibit certain bacterial growths using a solution or an agent that may be of a lower pH range. Acidic solution can lead to the death of microorganisms. Also, using alkaline solutions may be intolerant to certain microorganisms and will cause damage to these organisms and thus will inhibit its growth. If microbial cells are in a solution in which the concentration of solutes are higher than what's found in the cell, cellular water will pass out of the cytoplasmic membrane in the direction of the high solute concentration. During the loss of water, the cytoplasmic membrane will basically collapse. It collapses away from the cell wall. Remember, in bacteria, these microorganisms have both a cell membrane and a cell wall. If the cell membrane collapses from the cell wall, it's called plasmolysis, and basically this can cause or lead to the destruction of the microorganism. So another way to be able to restrict the growth is to eliminate or to alter the physical requirement of osmotic pressure. Hypertonic environments, increased salt or sugar, cause plasmolysis. Cytoplasmic membrane again will collapse from the cell wall. Now this is not true of all microorganisms. Certainly there are microorganisms that can live in hypertonic environments, but you can imagine organisms that typically want to infect the human system can't tolerate these osmotic pressure extremes because we can't tolerate these osmotic extremes. And if the organism can live inside of us, then basically that would tell us that these osmotic pressure changes can affect microorganisms. Extreme and or obligate halophiles basically require high osmotic pressure. Extreme halophiles, sometimes referred to as obligate halophiles, basically are organisms that have so adapted to high salt concentration, as high as 30% of them require them for growth. Now this is important because back in the days of yore, salt was used to help preserve meat products because microorganisms that typically led to food spoilage couldn't survive in these highly osmotic areas, and so that was found to be a great preservative for certain foods. Now you can see in this illustration where it points out, again, this is the typical arrangement, physical characteristics that we see in microorganism where you have a plasma membrane and a cell wall. Place this in a hypertonic solution, sodium chloride, 10% sodium chloride solution. Water leaves from the inside of the cell, moves to the outside. The cell membrane will shrivel up, basically collapse away from the cell wall, and thus stop or inhibit and or kill the microorganism. Facultative halophiles basically do not require high salt concentration, but they are able to grow at salt concentrations as high as 15%, and they tend to inhibit the growth of many other bacteria. The osmotic effects are roughly related to the number of molecules in a given volume solution for a given weight, volume. Therefore, sodium chloride, for example, is more effective than sucrose is for particular organisms. Under unusually low hypotonic osmotic pressures, not as much sodium as the solute in the solution, such as in distilled water, water will tend to enter the cell. Now you can see a similar process take place where too much water can actually enter into the cell, enter through the cell membrane, and cause a lysis or a rupture of the cell based upon an increased amount of water that enters into these cells. But facultative halophiles basically can tolerate high osmotic pressure. Now, besides water, this is which is needed as a nutrient, it must be in solution in order to be used. Carbon is the primary requirement for growth and the structural backbone of living matter. It's the organic substance. Those of you who studied organic chemistry can appreciate these this fact. Its valence is 4 and allows it to be used in constructing complicated organic molecules, sources of carbon and carbon dioxide, or other organic materials. Carbon, structural organic molecule, it's an energy source. Chemoheterotrophs use organic carbon sources. Autotrophs use CO2, still using carbon but just in a different source. Some organisms use other substances. Sorry, hang on just a second. So some organisms use proteinaceous material, nitrogen source. Others use ammonia. Now remember, in our system, when we deaminate proteins, we can't use the ammonia. Ammonia in our system is toxic. Ammonia in our system has to go to the liver, has to be broken down in the liver, transported, added, changed into urea, and then excreted out of the kidneys. But there are organisms that exist, microorganisms that exist out there that use ammonium or nitric acid or nitric ions in their systems as a source of energy. A few bacteria and cyanobacteria, which we'll study in more detail in some later chapters, able to use gaseous nitrogen directly from the atmosphere. This process is referred to as nitrogen fixation. Some microorganisms, which you'll be studying later, Rhizomium is one of these organisms, and Bradyrhizomium are bacteria that in symbiosis with leguminous plants. You may be familiar with certain plants are utilized in agricultural markets that help fix nitrogen or bring nitrogen into the soil, which helps to fertilize the soil, which helps for the use of growth for crops. So it's utilized in an agriculture as well, these bacteria that are able to do nitrogen fixation. Sulfur can also be used as a source. Again, these aren't substances we typically think about for the biological system to be used, but for microorganisms, sulfur can be a source. Sulfate ions, hydrogen sulfide, sulfur-containing amino acids can be utilized. Phosphorus, another agent that can be used. Phosphate ion can be converted, utilizes energy in many microorganisms. Nitrogen, sulfur basically are required to synthesize proteins. DNA, RNA, and ATP require nitrogen and phosphorus. There are trace elements as well that may be a part of the physical requirements for certain microorganisms. Certain or particular types of minerals that can be used. Iron, copper, molybdenum, and zinc, referred to as trace elements. You should know the trace elements, not that many of them. Although sometimes added to laboratory mediums, so you'll see them added to various culture plates to make specific microorganisms start to grow. Again, you can characterize or identify particular microorganisms based on what their energy requirement might be. And so even though sometimes they're added to these mediums, they usually are assumed to be naturally present in water and other medium components. Again, nitrogen, sulfur, phosphorus, these are various types of structures that can be utilized as energy sources for particular bacteria. The trace elements, again, inorganic elements required in small amounts. I reviewed those for you already. You should be somewhat familiar with the trace elements. Then oxygen. Microbes that use molecular oxygen are considered to be aerobes. If oxygen is an absolute requirement, they are referred to as obligate aerobes. Facultative anaerobes can use oxygen when it's present, but continue to use fermentation, which we've already discussed about, or anaerobic respiration when it's not available. But facultative anaerobes grow more efficiently aerobically than they do anaerobically, and we'll see this again inside the human system for microorganisms. They can live out in the environment even in the absence of oxygen. However, when they get into the human system, infect human cells that are more oxygen dependent, we'll see that these microorganisms can grow very efficiently. Obligate anaerobes. Now, these are bacteria that are totally unable to live in the presence of oxygen for growth, usually find oxygen to be toxic. So hydrogen atoms of the electron transport chain may pass or may be passed to oxygen. They form a toxic hydrogen peroxide. Now, hydrogen peroxide typically is very caustic to our cells. Aerobic organisms usually produce catalase. This is an enzyme that can break down hydrogen peroxide to water and oxygen. We see that in the biological system to be able to render hydrogen peroxide harmless when generated. Anaerobes usually lack the catalase that's able to do that. And then there are aerotolerant anaerobes. They cannot use oxygen for growth, but they can tolerate it very well. They will typically grow on surfaces of solid mediums without any kind of special technique or cultivation, but they're less oxygen tolerant than anaerobes. Common example of an aerotolerant anaerobes are basically bacteria that ferment carbohydrates to lactic acid, and the process again referred to as fermentation, as we see in the production of certain foods, alcohols, cheeses, that sort of thing. A few bacteria, microaerophilic organisms, meaning that they'll grow in oxygen concentrations lower than what we see typically found in air. Now they are aerobic, however, basically in a sense they require oxygen, but again it's at much lower, only at lower concentrations of oxygen than what we typically see found in our environment. They are probably usually sensitive to superoxides or free radicals or peroxides, which typically were produced in oxygen-rich conditions. And these are just the different categories. Again, oxygen-required obligate aerobes, facultative anaerobes, obligate anaerobes, aerotolerant anaerobes, and microaerophiles basically can exist in the presence of oxygen. Now, oxygen can come in toxic forms. Even though we breathe oxygen, there are some forms of oxygen that can be quite toxic. Oxygen can exist in a singlet oxygen, normal molecule of O2 that's been boosted to a high energy state and is extremely reactive. Superoxide free radicals are formed from small amounts of aerobic organisms, and they are so toxic that bacteria must neutralize them with an enzyme. The enzyme is superoxide mutase. This particular type of enzyme utilized can neutralize these superoxides. There's hydroxyl free radical formed in the cytoplasm from ionizing radiation, again, which supercharges these particular types of radicals, these free radicals. Byproduct is aerobic respiration, probably the most reactive form. And this is just kind of a review of those particular structures. Now, organic growth factors. Organic growth factors are organic compounds such as vitamins, amino acids, pyrimidines, which we'll talk about when we talk about the nuclear structure and genetic material, pyrimidines, which are needed for life, but also given organism is unable to synthesize, and so it has to be obtained from organic compounds. This is an organic growth factor. Vitamins, amino acids, purines, and pyrimidines, important. You should be familiar with these organic growth factors, again obtained from the environment. Now, there are certain culture mediums that we can manipulate or utilize and can be utilized to be able to help specific bacteria grow. And we want to utilize these culture mediums because they will help us to identify organisms based on the substances we use to make them to grow. As I alluded to earlier, given the byproducts that are produced by certain bacteria, it helps us to identify these microorganisms. When we can identify these microorganisms, if we plate them or culture them in a medium that will facilitate their growth, it helps us to be able to readily identify them. So a culture medium is basically any nutrient material prepared for growth of a bacteria in a laboratory, and it's basically called a culture medium. Microbes growing in a container of a culture medium is basically referred to as a culture. When microbes are added to initiate growth, then that's referred to as an inoculum. Now, those of you who have already done your early laboratory assignments probably can appreciate the little video that goes through some of the steps of establishing a culture medium, where actually microbes are cultured onto a slide or onto a medium, a culture medium, and you can see the growth of these various microorganisms. I'll have to check your new lab book. The lab book that I actually had wanted for the class actually has several nice illustrations clearly demonstrating a culture medium, the various types of culture mediums, and then also being able to demonstrate the microorganisms when they're inoculated or when they've been added to this growth medium, and then what their appearance would be, say, at a 24-hour, 48-hour, or 72-hour mark. If you want to ensure that the culture will contain only the microorganisms that were originally added to the medium and or their offsprings, the medium must then, or it must start off being initially sterile. Now, there are concepts of culturing microorganisms. Remember, when you open up a culture plate and you put a bacteria on that culture plate, especially if you use it by the appropriate technique and you culture it, if you leave it open to the air, there are microorganisms in the air that will also settle on the culture plate, and it may alter the different types of bacteria that may be growing. So you need to do this in a process that's relatively aseptic. In other words, you take an inoculant or a specimen of the bacteria that you want on the plate. The plate is upside down, face down, so that the cover is actually sitting on the surface and the culture medium is actually on the top. And when you pick it up, you flip it over, put on the bacteria, and flip it back down, so it limits its exposure to the environment. So hopefully no contaminants will fall onto the culture plate. And then it's placed in a incubation area, whether that's at a warmer temperature, colder temperature, medium temperature, whatever, and the microorganisms will virtually grow on those plates. When a solid medium is required, a solidifying agent is added. It's referred to as an agar. An agar basically is a polysaccharide derived from seaweed. Certain nutrients are added to this seaweed or this agar. It kind of looks like Jell-O. It's a little thicker consistency than Jell-O. It will melt at about boiling point where it remains in a liquid until the temperature drops to a certain degree. It's an excellent medium for growing particular types of microorganisms. Complex polysaccharide is what makes it up. Uses solidifying agent in the culture medium. Typically it's placed in a petri dish, a little clear plastic dish. Again, when you do your lab exercises, you'll be able to see petri dishes and how they're utilized. Generally, these substances that are placed in the agar dish are not metabolized by microorganisms, liquefies at about 100 degrees, and then solidifies at about 40. Culture mediums typically have nutrients. These nutrients are prepared for specific microbial growth. So you typically, if you're trying to culture out a microorganism, you would know what the requirements were for that microorganism for growth to be able to take place. And then the inoculum, the introduction of microbes into the medium, and then a culture, basically microbes growing in and on the culture medium. The culture medium, chemically defined media, exact chemical composition is known. The complex medium are extracts or digested yeast meat. So these are special or complex types of media that can be used to grow certain types of microorganisms. You can add specific nutrients into this culture medium. They have nutrient broths, and then nutrient agars can be used. Again, special medium to grow special organisms, and again uses specific types of substances. And this is the composition of particular types of nutrients, chemically defined nutrient agars, which contain certain chemical constituents, glucose, ammonium, sodium chloride, magnesium, so forth. Then composition nutrients, which may have peptones, beef extracts, sodium chloride, agars, water, etc. For anaerobic growth mediums and methods, you get obligate anaerobes. Bacteria often require reducing medium for isolation. Because oxygen may be lethal for some of these organisms, the media typically contains ingredients that will basically chemically combine to dissolve the oxygen. Sodium thioglycolate is typically one of those substances that's used. Thioglycolate basically will deplete oxygen contents in culture mediums. Obligate aerobes may be grown on the surface of these solid mediums, which basically are oxygen-free atmospheres, but it's generated based upon a chemical reaction. Now, there are also enzymes, oxidase, which basically can be added to growth median. So if it's possible to handle anaerobic organisms in new techniques when you utilize this enzyme, it's added to the growth median and basically will transform a Petri plate into an oxyplate or a self-contained anaerobic chamber, again where the oxygen would be removed. The reducing medium contains chemicals, thioglycolate or oxirase. This basically will combine with O2, heat it appropriately, and then it drives off the O2, so it will not be in the median. This is just an illustration out of your textbook that shows an anaerobic culture mechanism. Again, basically what I would want you to be able to appreciate from this is that in this particular type of mechanism, this anaerobic culture is set up where chemically and enzymatically oxygen is removed from that environment, and anaerobic organisms would be able to effectively grow. Now, special culture techniques. There are a few bacteria that never been grown successfully on artificial laboratory medium. Leprosy is a good example of that. A mycobacterium, similar to tuberculosis. Syphilis or other organism of example has not been successfully grown or an artificial laboratory medium. Typically needs a live organism to be able to survive and to be able to be demonstrated for examination. Obligate intracellular parasites. Obligate intracellular parasites have to live inside a cell, living cell. Rickettsia and chlamydia also do not ordinarily grow in artificial mediums. They, like viruses, require a living host cell to be able to survive. Carbon dioxide incubators, candle jars, plastic bags, self-contained chemical gas generators are used to grow bacteria with special CO2 concentration requirements. Capnophils basically are microbes that grow better at high CO2 concentrations. And I haven't seen this utilized in a while, but it's probably still out there. The candle jar, where basically see a lid, you see the Petri dishes that are stacked inside. Now, if you notice, and those of you who haven't seen the slide and aren't very familiar with Petri dishes, if you look very closely, the auger is basically on the lid portion, or what appears to be the lid portion of this Petri dish. And basically you can lift it out of the lid, actually is on the bottom, and you lift it out of the lid, smear it with the bacteria, flip it down on the lid. But in this environment they basically use the flame to drive off all the oxygen, to utilize all the oxygen that's inside the jar, and then you have an anaerobic container. It also will increase the level of CO2 inside the jar, and so you have a CO2 enriched environment, which works extremely well for bacteria that require CO2 as one of the physical factors for growth. You also can have a chemical packet, a CO2 packet, which has a gas generator in it. Again, you see the Petri dish. You see the Petri dish inside the bag. You typically will break this generator. It's a chemical compound that you can utilize. You break the test tube, which is inside the bag. Again, it generates CO2 and drives off O2. So now this brings up two key definitions: selective media and differential media. Selective media are designed to suppress the growth of unwanted bacteria, encourages the growth and desired microorganisms. Antibiotics, high concentrations of salt, high acidity might be used. Again, the point here is you want to design or suppress the growth of unwanted bacteria, and it encourages the growth of desired microorganisms. Differential medians are much easier to distinguish colonies and desired microorganisms from other colonies growing on the same plate. Colonies have different colors. They cause different changes to occur in the surrounding median, and sometimes selective and sometimes differential functions are combined in one median to be able to get specific growth. To use a selective median, you may want to suppress the growth of microorganisms and encourage the growth of other microorganisms to occur. Differential medians makes it easy to distinguish the colonies different from others. And so you can see basically from these slides the differences in the two types of medians and how they can help you to identify particular microbes. Now, enrichment medians, special median that you utilize to try to promote growth. Because bacteria may be present in only small numbers, it might be missed. So because of this, you want to isolate a particular bacteria that has an unusual physiologic type, and sometimes it's necessary to actually add substances to a median that promote the growth of this particular type of organism that you don't have enough of the numbers for. And so you use what's referred to as an enrichment median. Conditions are set so you get the sole nutrients that are designed to increase the small numbers of a certain organism till they can grow to a detectable level. Remember, some of the bacteria we need to study. It may not be easy to grow it in the environment, and so you have to set up conditions so we can grow more of the microorganisms, thus to be able to study them more effectively. Enrichment medians basically encourages the growth of desired microbes, assumes that soil samples contain a few phenol-degrading type of bacteria and thousands of other bacteria. You want to quickly get out the other bacteria. You want to be able to differentiate those, so you may inoculate phenol-containing cultural medium with the soil and incubate it. You would transfer it serially to different types of flasks that would contain the enrichment substance that that particular bacteria would like to be able to grow. Probably the most common method that we see in a laboratory study or laboratory setting to be able to identify microorganisms would be the streak plate method. Now, in the streak plate method, you get a sterile inoculating needle, and again, you'll have to appreciate this from the lab. Basically, you get a sterile inoculating needle or a loop. It's basically just a straight cylindrical wire loop, little eyelet, little circle at the top of it. You basically would dip that into a bacterial substance, whether that's a urine specimen, whether that's a sputum specimen, whether it's even from a blood specimen, though that would be a little bit different. But you dip it into a bacterial source, and then you actually smear it along that agar plate. And again, the technique, the lid is down. The bottom of the plate actually looks like the lid. It's turned upside down. You flip the plate up. You take your inoculating, I did that backwards. You inoculate. You then smear it on the slide, and then you put it back down into the cover, and it'll grow. Now, there are specific steps involved in that. In your lab, when you watch the videos on the lab, it shows you how to do the streak plate method. But a sterile inoculating needle is typically dipped into a mixed culture. It's streaked on the plate over the surface of the nutrient medium, then the last cell rubbed out from the needle where it's wide enough apart to grow. And then you get masses of colonies will grow on these particular types of culture plates. Now, for a pure culture contains only one species or strain. Colony is basically a population of cells arising from a single cell or spore or a group of attached cells. A colony is often called a colony forming unit, or CFU. And this is a streak plate. Now, when you initially do a streak plate, as you can see the pattern, the loop is basically taken. If the arrow is the tip of the loop, you make these little Z lines, or zigzag lines across the top. You turn the plate, and you'll do it on the other side, and you'll turn the plate again, and you'll do it from the other direction. And this is the resulting bacterial growth that you'll see on the left. And then you may want to do a more thorough differentiation. There are different types of bacteria that are growing here. You can tell that based upon the color. See how some of them are a little lighter yellow, and some of them are dark red. You may then want to take this different inoculating loop and a different culture plate and try to culture out specifically just this organism, or culture out specifically this one. You take the loop, take a specimen of this, put it on a different plate, take a new loop or a new specimen obtainer, take just a specimen of this and put it on another plate, and then see what grows. This is why cultures take several days to come back. Cultures probably take 72 hours, sometimes even longer, because you may get a number of microorganisms here, some of which have disease-causing capabilities and some do not, but you'd have to differentiate those. This is why the gram stain is so important. The gram stain, when you come in sick, let's say you have a pneumonia, you can produce a sputum specimen. If I want to culture that specimen, it'll take me 72 hours to find out what type of bacteria is in your lung. If If I do a gram stain, I'll immediately know if it's gram negative or gram positive. The treatment that I use to treat that pneumonia will be based on the type of organism that's present. If it's gram negative, I'm starting you on 2 big-time drugs. If it's gram positive, I'll probably start you on 1. And so you gauge your therapy based upon gram negative, gram positive. 3 days later, I know specifically I have a Bacteroides type of infection in my lungs, which requires a specific antibiotic, and I can be a little more specific in my treatment. Or I have a Staph aureus pneumonia. Again, I find that out 3 days later, and I can gauge my therapy. But this way, just by the gram stain, I can immediately start therapy, because the streak plate method, growing out